Hello students, in continuation with the Recombinant DNA Technology series that we have been doing, today I would be talking about the visualization of DNA and DNA fragments. The specific objectives for this lecture would be to understand the visualization of DNA or the restriction digested DNA fragments, to understand the setting up of the agarose gel electrophoresis, and to understand the purification of DNA fragments from agarose gels. So, just to review, we have talked about the isolation of DNA, both the plasmid as well as the genomic DNA and the DNA from bacteriophages. And we have also talked about the manipulation of these isolated DNAs. We started with the different enzymes that can be used to manipulate the genomic DNA as well as the plasmid DNA so that they can be combined together to form the recombinant DNA. In these series, we started with the restriction endonucleases. We have talked about nucleases, that is the enzymes that cut the DNA molecule. Because an essential aspect of recombinant DNA technology is that the vector, which will be the vehicle, as well as our gene of interest should be cut and then religated so that the recombinant DNA can be formed. So, when we digest the DNA or when we isolate the DNA, the next thing that comes up is to visualize whether there is DNA or whether it has cut or what are the different sizes of the DNA fragments that we have obtained. So, for any further manipulation or for any further step of the recombinant DNA technology, at first we need to see or visualize this particular DNA or DNA fragments. So, I talk about that in this particular lecture. The references which is used primarily for the educational purpose is the book T.A. Brown, The Gene Cloning and DNA Analysis and Introduction, the 6th edition, and it is entirely used for the educational purpose. Now, whether the intact DNA or the restriction digested DNA fragments, a technique is required to see this DNA and as well as analyze the result of the restriction endonuclease cleavage. A restriction digest results in a number of DNA fragments, the sizes of which depend on the exact positions of the recognition sequences for the endonuclease in the original molecule. So, again I review from the previous lecture in which the restriction digestion was taken up, that when a restriction enzyme cuts the DNA, it cuts at specific sites. Now, there can be more than one site which is present in a DNA molecule. Now, based upon the location of these sites, the DNA fragments would be obtained. For example, in this case, you can see that there is a DNA in which there are 3 restriction enzymes which are given. And the 3 restriction enzymes has a number of sites. For example, BGL has 6 sites. Based upon that, if you can see the lower part of the figure, you can see that there are different sized fragments that are obtained. So, the 6 sites of BGL are not equally placed, and therefore, when we cut this DNA molecule with this particular enzyme, then you obtain such fragments. The sizes of the fragments are also given. Now, when we have restriction digested a plasmid or a genomic DNA and we obtained such fragments, then you know how to find out that what are the sizes of the DNA fragments or how to isolate or locate where our gene of interest is lying. So, based upon these digests, we can separate them. Now, the separation of molecules can be done by what is called as gel electrophoresis. Now, electrophoresis is a technique that uses differences in electric charge to separate the molecules in a mixture. So, what we use is an electric current which will separate the different molecules. Now, as we all know, DNA molecules have negative charge, and so when they are placed in the electric field, they migrate forward to the positive pole. So, again you can see here, so it is such a kind of a setup. We will go in detail about the setup of this particular thing, but as far as the electrophoresis is concerned, you can see that there are 2 electrodes and at one end the DNA is loaded in a well. Now, when the electric current is passed, then the DNA from the negative side will start moving towards the positive side because it itself is negatively charged. So, DNA migrates towards the positive end, and this movement is we can harness to even separate the different DNA molecules. Now, if we have isolated and intact, if we just want to visualize the genomic or plasmid DNA, which will act as a source of our DNA, then we can just load the complete, the direct, whatever isolated DNA solution is, we can just load it in this particular well and we can visualize it under, and we will see how we are able to see it, but we can just run the complete DNA in such a setup. And when it is a digested fragment product, then the digested product can also be loaded in this well and then run in a same manner. Because when an electric field is passed in such a setup, then the negatively charged DNA will move towards the positively charged electrode. So that is the basic concept of electrophoresis, which will be harnessed to in the agarose gel electrophoresis, which we talk about. The rate of the migration of a molecule depends upon two factors, its shape and its charge to mass ratio. Now, the matrix which we use here in electrophoresis for DNA is generally agarose. Now, what happens is when a molecule is moving in a matrix, then this movement or the speed of movement will depend upon the shape of the molecule, because bigger the shape is, slower the movement would be, and particularly then the charge to mass ratio. Now, in here the charge is same because DNA is negatively charged, so it will entirely move according to its shape, or what we call here as the size of the DNA molecule, which is measured as the length of the base pairs. So, but in other cases, if we are running different molecules, for example, proteins, then the charge to mass ratio will also vary, but we will have to neutralize it to run it on particular gels. But we are talking about the DNA here, so what majorly affects the movement of the DNA in agarose gel electrophoresis is the size of the DNA molecule. Now, gel electrophoresis separates the DNA molecules according to their size. So, size basically means the length of the base pairs. Now, because it is gel, that gel is made up of agarose or polyacrylamide or a mixture of the two. It comprises a complex network of pores through which the DNA molecules must travel to reach the positive electrode. So, as I said, the setup contained two electrodes, and we know that we have to run the gel from one end to the other end. Now, that running is through this matrix, which can be made up of these substances such as agarose, polyacrylamide, or even a mixture of the two. Now, this agarose under room temperature, the agarose solution solidifies. It's a semi-solid kind of a thing, and it gives rise to a matrix which has pores in it. Now, the DNA will move through these pores and will move from one end to the other. So, this network of pores through which the DNA molecules will travel to reach the positive electrode. And definitely, as you can imagine, that the speed of the molecule will depend upon whether or how fast it can pass through the pore. Now, pore will have a limited size, and therefore, the movement will depend upon the DNA size. So, because the pore is constant, the size of the pore is constant, larger the DNA molecule, slowly it will pass through, smaller the DNA molecule, it will pass through very fast. So, that is basically the principle behind the separation of different size fragments inside this gel matrix in the agarose gel electrophoresis. So, this is just reflected here, that if you load the DNA, now in this case the DNA is a digested product which contains differently sized fragments, as we saw in the slide where, for example, BGL had 6 sites, and we could see that the 6 fragments were of different sizes. So, if we load the digested product in the well and then apply the electric current, then this particular DNA has to pass through this gel. Now, the gel is containing the pores. So, as I said that the smaller molecules will run faster, whereas the larger molecules will run slower, because they have to pass through a constant sized pores. So, again as shown in the lower half of the figure, the fragments will separate out based upon the size. So, this is a very useful technique to separate the DNA fragments based upon their size, that is the length of the bases. Now, the composition of the gel determines the sizes of the DNA molecule, that is the length in base pairs that can be separated. Now, again this is a very important aspect because the DNA is of varying sizes. If we look at a plasmid DNA, when we talked about cloning vectors, then the plasmid DNA were ranging in size from as small as 500, 600, 200, 2000 base pairs. For example, the most common plasmid such as PUC18 is 2.6 kb in size. So, it was basically ranging from the kbs. But when we talk about the genomic DNA, the size is in mega base pairs. The length of the base pairs is in mega base pairs. So, when you have to run it in a gel, then obviously you know that there are different sized fragments. And this different sized fragments will have to pass through the pores. So, if we vary the size of the pores, then the resolution of these fragments or these different types of DNA, we can have it in a better way. Now, varying the size of the pores basically means changing the concentration of the agarose solution. Because lower the concentration of the solution, bigger will be the pores. Higher the concentration of the solutions, smaller will be the pores. If the pores are smaller or you use a higher concentration of gel, then the resolution will be better. But if you use lower concentration of gel where the pore size are bigger, the resolution will be less. So, I hope you understand what I mean by the resolution is the distinction between the different sized fragments. So, if the pore size is bigger, for example, the 100, 200, 300, 400 base pairs will all pass through the pore and you will not be able to differentiate between the 100, 200, 300 or 400 base pair sizes. But if you keep lowering the size of the pore, then probably 100 base pair is able to pass, but 200 base pair is not able to pass or will pass through it slowly. And therefore, the 100 and 200 base pairs will differentiate on the gel. So, that relates to that the concentration of the agarose solution will determine the size of the pore, and the size of the pore will determine the resolution of the DNA fragments, or what is the size of the DNA molecules that can be separated on that particular gel in a better manner. For example, a 0.5 centimeter thick slab of 0.5% agarose, which has relatively large pores, would be used for molecules in the size ranges of 1 to 30 kbs, showing molecules of 10 and 12 kb to be clearly distinguished. So, if you have got a thicker slab and if you have got a concentration which is lower, then you will probably have a larger pore where the resolution would be like 10 and 12 kb can be clearly distinguished. But if the fragments are of base pair, differing in base pair sizes, then probably you are not able to distinguish them and they tend to move together. Similarly, if you use a very thin, for example, 40% polyacrylamide gel with extremely small pores, it would be used to separate much smaller DNA molecules in the range of 1 to 300 base pairs and could even distinguish molecules differing in length by just a single nucleotide. So, such a kind of resolution that even a single nucleotide base pair change is detected as separate fragments on the gel can be attained if you use a higher concentration of a gel, and therefore you use a material which can form such higher concentration of the gels and a very, very thin gel. Such gels are used for the sequencing purposes, because now the sequencing has been automated and there are many different kinds of sequencing methods that have been developed. But the initial, the basic Sanger sequencing method, or which used to use manual running of the gels, such gels were used, because there the distinction in the fragments have to be in the range of just a single nucleotide, because then only you can find out the sequence of the particular fragment. So, that explains that what will be the choice of the material as well as the concentration of the gel as well as the thickness of the gel which you need to use will depend upon the size that you need to fractionate, what you are looking at. and how much resolution do you want. So always keep that in mind. So we will look at the setup for the preparation of the agarose gel solutions. So how do we do it in a lab? Because agarose gel electrophoresis is a very routine technique which is used in any lab that works on the DNA or the RNA molecules. So nucleic acid and even the polyacrylamide gel electrophoresis is used for the separation of proteins. So what is basically done is that the agarose solution is made. So as I said, the concentration of the solution will depend upon what do you want to separate and what do you want to resolve. So the first thing is to weigh the amount of, so generally 0.8% to 1% agarose gel solutions are made to separate the genomic DNA or to detect or visualize the genomic DNA, plasmid DNA or the restriction DNA fragments routinely. So 0.8% to 1% agarose solution. So the agarose solution is basically weighed and then it is dissolved in the buffer. So we can use two kinds of buffer, which is a TBE or TAE, which is tris borate edita or tris acetate edita. These are the buffers which is needed and the gel needs to be made in this. A TB appears to be a little better because it works for longer time and it is more resistant to heat. And when we are running the gel in an electric current, then the heat is produced and the buffer tends to be heated. So TB is more resistant to that, so it gives a little edge, but it doesn't make a difference whatever buffer you use. The thing which one has to remember is that the buffer which is used to make the gel, you run the gel in that buffer only. That is the only thing which needs to be remembered, otherwise either of the buffers can be used. So TBE for example, so 1% solution you will weigh 1 gram of agarose and you will add 100 ml of TBE solution. As for any other routine chemicals, the stock solutions for these buffers can be made and working solution can also be used. So working is generally 1x or 0.5x TBE and stock solutions of 10x TBE can be made and stored, and at the time of need the solutions or buffers can be diluted. So the next thing is when you need to dissolve the agarose completely, and which is done by microwaving it for 2-3 minutes. So until it comes to boil and it becomes a transparent solution. You cool it for some time because it has to be, you do not cool it to the room temperature because then it will solidify. But you cool it a bit because you have to pour it into a casting tray to cast the gel. At this time, for quicker and easier purpose, one can add the stain inside it, the stain or the dye which is going to stain the DNA eventually. For example, in this case, ETBR or the ethidium bromide is added. Ethidium bromide stock is made 10 mg per ml and the working is 0.2 to 0.5 microgram per ml solution. So, while it is cooling, the ETBR can be added in here. Now, a very important precautionary method that needs to be taken is, and the knowledge of it should be there, is that ethidium bromide is a very powerful mutagen. Therefore, the handling of ETBR should be very careful. One has to use the gloves, the lab coat, and in fact the goggles for using such a harmful chemical reagent. So ETBR is basically added at this, can be added at this point. If it is not added at this point, then after running the gel will have to be stained for half an hour to 45 minutes in an ETBR solution put separately. So it can be done in the either way, but as I said, the precaution needs to be taken and one has to be cautious about the fact not to come in direct contact with ETBR because it's a very potential and a powerful mutagen. So this is the agarose gel electrophoresis apparatus. So you need a casting tray and you need a tank in which the gel will be cast. Then the combs are there which will make the wells where the DNA sample will be loaded. This casting tray, the gel and with the wells will be put in a tank where it will be submerged inside the buffer. Now, variety of agarose gel electrophoresis apparatus comes. It depends upon, how many samples do you want to run. You have like 8 well machines, you have 12 well machines, you have 20 well machines, 24 well machines. So, you have a lot of different companies provide these things. But these are the basic things which are required. So, you will have then the electrodes and you will have a voltage source. So, these are the basic things which is there. So, in the casting tray, we put the comb and we pour the solution of the agarose containing the ethidium bromide. So, this is put like this. So, it has to be sealed from all the 4 sides so that it does not leak. And on the top, you can see that a well comb is put in and the agarose gel is carefully poured into the casting tray. Now, the gel does not have to be very, very hot. One has to remember that, but it should not be that cold that it will give rise to lumps. Bubbles should be avoided, and in fact, any bubbles should be pricked with can be pricked with a tip. Normal tip also, which can be there. After putting this, then the agarose gel, after it sets, it takes around half an hour to set the gel. So, when the gel is completely set, the well comb is removed and the gel is put inside the tank and the buffer is added. So, again, the buffer which is to be used should be the same as in which the agarose gel solution was made. So, whether you use TBE or TAE does not make a difference, but the solution has to be the same. So, it is submerged inside there and then your samples are loaded. Now, you can note here that the samples are blue in color and they are carefully loaded with the pipette, where the pipette tip is lowered down inside the well, and in one go the sample is released into the well. Now, when you isolate the DNA or when you run a restriction digestion reaction mixture, then it is finally dissolved in water. That is what we talked about. So, but when you are loading it onto the gel, because everything is transparent in color, it becomes very difficult to visualize your DNA solution. If, for example, the DNA was just transparent in color inside the pipette, when you load it into the well, you are not able to track it. So, for tracking the DNA and for making it easier to visualize, we add the loading gel buffer in it. The loading gel buffer contains 2 important components. One is that dye which is staining it blue. For example, in here bromophenol blue is used. There are other dyes that can also be used in the gel loading buffer, but the most common one is the bromophenol blue. And another thing which is either the sucrose or the glycerol, because sucrose or glycerol will give the density to the DNA sample so that it stays inside the well and does not diffuse into the adjoining buffer, which is the the buffer which is there. So, the loading buffer is added to the DNA sample which needs to be visualized or to the DNA fragments which needs to be seen onto the gel. So, the loading buffer serves 2 purposes. It provides a visible dye that helps with gel loading and allows one to gauge how far the DNA has migrated. It contains a high percentage of glycerol or sucrose that increases the density of DNA sample, causing it to settle at the bottom of the gel instead of diffusing into the buffer. So, again the loading buffer can be made at a higher concentration, usually 6x, and 1x is used in the each DNA sample. The electrodes are then put into the voltage source and the gel is run. The gel, now you can see that blue line, so that will help us track where our DNA samples is going. So, DNA is above it, depending upon the size it will be lying somewhere there, but we know that this dye, so when this dye runs around 3/4 of the gel, then it has sufficiently run and we can visualize it. So, the size of the DNA is approximately 200 to 300 base pairs, so that we can keep in mind that it does not just flow out of the well. Now, when the gel is run, we need to visualize it, and as I said, we have added the ethidium bromide which is routinely used to stain the DNA in agarose and polyacrylamide gel. The bands show the position of the different size classes of DNA fragment are clearly visible under ultraviolet irradiation after ETBR staining. So, when you stain it with the ETBR, so that is the gel that has run, and when you stain it, you are able to visualize these DNA molecules under the UV light. There are alternatives to ETBR because ETBR is a mutagen. There are, for example, cyber safe DNA gel stain or cyber green 1 nucleic acid gel stain, which are less harmful and the sensitivity is also higher. So, ETBR is commonly used, but now safer varieties of these stains are also available that one can purchase and use it safely. So, this is how a gel will look like in a over a UV transilluminator strain. So, this is a plasmid DNA isolation preparation, and in plasmid DNA you obtain 2 gels because there are at least 2 to 3 bands, because there are different conformations of the plasmid DNA which is present inside a cell. This is a total intact plasmid cell isolation from E. coli DNA. So, you can see the RNA smear. It was a crude method. So, you can see the RNA smear which has taken up a lot of stain, but it will eventually lose it, and the plasmid DNA molecules. There are 2 DNA molecules which are there. So, they can be separated based upon the fragment size also, because the fragment size you can measure by running it along with the DNA ladder. So, DNA ladder is the DNA molecule which has been digested and the sizes are already known. So, we know that there are so many base pairs and they are running at these positions. So, if we run our digested DNA fragments along with the ladder, then comparing it by the look of the eye, we can actually predict the sizes of the DNA fragments which are there. So, this, for example, there is an undigested DNA which is shown by the arrow, and there are 2 digested DNA fragments which can also be seen here. A better composition of the DNA, estimation of the size, is given by this formula, but generally what we do is we run it along with the DNA ladder and then we look at the size of the DNA fragments. So, that is how we visualize the DNA on the gel, whether it is genomic or plasmid DNA, or whether there are restriction DNA fragments, and we also measure the size of the DNA fragments by running it along with a ladder. Thank you.